So now you know a little bit more about cell competition. First, you saw the work of Malcolm about cell competition in fruit-like legs. Then we went and explained, you read this paper and answered some question about cell competition, and now you know that cell competition is very important, that it can help us to understand how you can go from a normal epithelial, suppose that this is a normal epithelial tissue, and how it become a cancerous epithelium, so that you have a tumor here growing, and how this tumor is killing the other cell, how it's outcompeting the other cells, and this transition between a normal and a cancerous epithelial is fundamental, because this is the best way to fight cancer, because it hasn't grown so much, it hasn't moved from the epithelium. So understanding cell competition is very important. And there are two groups. So one will study the difference in width and height during cell competition, that we will study in apical area. So how the tissue is getting, if the tissue during the cell competition, the height is changing, or the width is changing, and is also oscillating as described Malcolm, or is not changing, and why. And the other group, so check that you're in the right group, it will work on understanding the cell perimeter. So what happened with the cell perimeter? Do we have oscillations similar to the ones that Malcolm described for the area, or something different is going to happen? So for this class, I wanted, there will be two things that you have to do. So you already took the quiz. So now you go here in the section of cell competition, and you will have to hand in two things. One that it will be today at midnight, and the other one will be before next class. So what you have to do is to read a publication related to this topic, and I think the best way to start is to read the work that Malcolm, Jaden, and Chris did. And, and please, we read it hundreds of times, but if you see any typo, any, and if you have any suggestion, if you find something that there is something not clear, please write it down, and when you upload your comments. Uh, and I think this could be a good way to start, so you can start predicting what happened with the cell perimeter, what happened with the width and the height, depending on your project. So this is a good way to start. And in addition to that, to reading this and answer a few questions, I want that you also later look for more publication on this topic. So go to PubMed. Yeah, so publications in medicine. So you go PubMed, and then there you this is or Google Scholar, any of those that are useful. And if your topic is cell perimeter, so look what additional information we have related to the cell perimeter, what other people have studied about the cell perimeter, and you can send me the PDF, and you can skim the paper. And also, if you Mm, if your topic is the width, the changes in width and height, you can also look for the apical changes in width and height. Remember, if the cell is three-dimensional, we are measuring this region, the top, and here this. So we are studying the width. It will be this, and the height would be this. We are not studying it in a three dimension. We are studying the top of the cell. So remember, this word apical is important. So if you find any paper or anything that is interesting, send it to me. Even and if you cannot download it, send me the link, and I might be able to download it. I just want that you start looking for information so you can add it to your scientific poster. So to summarize, you have to read this paper, answer a few questions that I will post there, and you have to do it today. And if you find any typo or anything, please add it. Write like, I found that in this sentence I found this typo, and I will be very happy if you find many of those. In addition, for next week, you just have to find a few publications that talk about the topic of your research on cell perimeter or in apical cell width and height. And this will be next week. And again, you can upload the answer of the questions here, and you can upload the papers or the links of the papers here next week. So you have a week to look for few publications. And if you have any question or any doubt, just let me know, and I will answer it. Have a good day and see you.